Good day, the organizers, the coordinator, all other protocols are well observed. My name is Lawal Teslim Malabi, a research member at Professor Abdulhamid Biophysical and Computational Research Group at Ladoke Akintola University of Technology, Ogbomoso, Nigeria. I'm here to present a research findings on anti-SARS-CoV-2 activities of phytochemicals from Nigella sativa using molecular docking, molecular dynamics, and chemoinformatics approach. Here is my outline. Coronavirus disease is an infectious disease caused by coronavirus, which began its human invasion in the late 2019 and has been reported to have been originated from a city in China. And according to the World Health Organization, this disease has claimed over 7.6 million lives, and about 800 million persons were reported to have been infected since its outbreak in late 2019, according to the July 2023 World Health Organization report. Due to the novelty nature of this virus, the traditional method of drug designing, which is time and cost consuming, seems ineffective or inactive, as a novel and a quick approach is required. Hence, the need for drug repurposing and phytochemical screening to identify potential drug candidates against this deadly virus. It is very pertinent to understand the mechanism of action of any disease in order to have an insight about how to design a drug against it. This figure represents the prevalence of coronavirus in different parts of human organ system. As it is represented in this legend, the darker the blue color, the more prevalent the virus in the part of the body. So that shows that the upper airway, the mouth, lungs, gastrointestinal tract, urinary system, and immune system are the most vulnerable and the most infected part of human organ system, whereas liver, gallbladder, and pancreas, as well as skin and adipose tissue, are less infected. Coronavirus disease infected the host through its spike protein, and it enters through the host DPP4R using the angiotensin converting enzyme, and it attacks the macrophages through its antigen producing cell. So as a means of immune response, the immune response alerts the T cells through the T helper cells and secrete the cytokines such as interleukin and interferon in order to engulf the virus to prevent its further replication in the host cell. However, the much secretion or over-secretion of the cytokines, it's also detrimental to the host cells, such as NF and KB, which are pro-inflammatory, that could lead to another serious ailment, such as cancer. It is known to almost everybody that COVID-19 is one of the deadliest invention and pandemic in nature in the recent year, with over 7 million lives being claimed, though number of vaccines have been approved for emergency use by at least one regulatory authority. However, there are still some underlying comments about the efficacy of these vaccines, as well as equal and optimal uptake in all countries of the world, such as underdeveloped and developing countries. In addition, there is no single approved drug by the World Health Organization as cure for this disease, and more variant of this virus is surfacing in different parts of the world. Therefore, the need to search for natural products from plants that possess anti-COVID-19 properties cannot be overemphasized. One of the major reasons for our research is the challenge of immediate cure, considering the short time of this disease mortality. Also, the easy accessibility, cost-economic nature, and wide pharmacological properties of Nigella sativa make it more important to investigate the effect of its phytochemicals on this particular virus. Using natural products that have been extensively researched on, such as Nigella sativa, to possess numerous phytochemicals with pharmacological and therapeutic action in model control studies is of paramount importance. As stated earlier, due to the novelty nature of this disease, the common and the traditional method of drug design seems ineffective. Therefore, a quick and novel approach, which is the application of in silico approach as a means of developing drug to prevent this virus, cannot be overemphasized. So this research is aimed at investigating the inhibitory activities of phytochemicals from Nigella sativa against SARS-CoV-2 main proteins with PDB-IDCCLU7 using computer-aided drug designing approach. The objective of this work include investigation of the amino acid residue involved in the interaction of the ligand, that is the phytochemical, and the target receptor using interaction visualizing software. Also, to identify more effective compounds as potential drug candidates against this disease by investigating and comparing the inhibitory efficiency of the docked ligands against the target receptor with standard antiviral drugs. So in this case, the selected standard antiviral drug is remdesivir. In addition, to examine the selected phytochemical safety for human consumption by carrying out chemoinformatic screening such as absorption, distribution, metabolism, excretion, and toxicity, as well as oral bioavailability, bioactivities, and drug likeness of the identified lead phytochemicals and compare them with that of the standard antiviral drug. And lastly, to predict the stability of lead compounds and the movement of the atoms in a biological system as time-dependent function. How this work was done? The first thing that was done was reviewing the work of literature to identify the chemicals present in this particular plant, that is Nigella sativa, and retrieving them from molecule data bank such as PubChem and zinc data bank. So after retrieving these phytochemicals, they were treated by converting them to docking readable format. After which, the target receptor was also identified and retrieved from protein data bank, being treated in order to get rid of other impurities such as water that comes with it, which may intervene with the docking process. Having prepared the target receptor and the phytochemicals were then docked against the target receptor using autodock tools and autodock vinyl software in order to investigate their inhibitory potential and compare with that of the standard drug. So those phytochemicals with better inhibitory activities than that of the standard drug were further subjected to other computer-aided drug design analysis, so which is ADMET analysis, in order to investigate their safety for human consumption. And those phytochemicals with good ADMET properties were further subjected to drug likeness using mode inspiration internet software, server-based. So those phytochemicals with good ADMET analysis or ADMET property that has been investigated by using ADMET SAR2 online server and that of the drug likeness using mole inspiration were further subjected to bioavailability tests, oral bioactivities, and their binding mode and molecular interactions were visualized in order to see the amino acids that were involved using Discovery Studio software. And those leads identified from number of phytochemicals earlier retrieved were then subjected to molecular dynamic simulation using GROMACS in order to investigate their behavior and that of the atom in a biological system, as molecular dynamic simulation mimic the exact biological system. And the results obtained from molecular dynamics simulation were used to plot a trajectory, a graph-like trajectory, to in order to visualize how these molecules behave. This was done by using XM-Grace software. Computer-aided drug design is one of the novel drug designing approach, and the fastest growing approach. And this research can be done in two ways. It could either be structural based or ligand based. So for this work, the structural based approach was employed. This represents the SARS-CoV-2 target receptor and the active site, as these main proteins have two active sites. That is, the main active site where the native ligand was bonded to, and that of the allosteric site in which some of the identified phytochemicals and the standard drug bonded into. This represents the pictorial image of black seed, also known as Nigella sativa, that is its plant, the flower, and the seeds. There are enormous pharmacological activities being identified by previous researchers and ascribed to Nigella sativa. So these activities include antibacterial, antifungal, anti-inflammatory, anti-cancer, antimicrobial, and antiviral. So these enormous pharmacological activities are one of the reasons why this particular plant was chosen to be screened against SARS-CoV-2 main proteins. The results obtained after the molecular docking and are presented as such. The red highlighted phytochemicals were the red flag. What do I mean by that? They were the one with lesser binding affinity as compared to the standard drug, with a binding affinity of 7.6 kilocalorie per mole. So those phytochemicals with less than 7.6 kilocal per mole were considered to be discarded, and those with higher binding affinity than the standard drug were considered for further analysis. And those part of those phytochemicals are the nigellidine, ederagelin, and trigelytin. So trigelytin, ederagelin, nigellidine, as well as the remdesivir, which is the standard drug, were further subjected to the ADMET analysis in order to investigate their safety for human consumption. So going by this result, the red highlighted phytochemical, which is trigelytin, have poor ADMET properties, which specifically it inhibits the human either agogo related gene that is related to potassium channel blockage. So as it is being stated by work of literature, that any molecule that inhibits this particular property should not be considered further for drug designing analysis. So hence trigelytin are being considered to be discarded, and ederagelin, nigellidine will proceed to the next drug design analysis, which is drug likeness analysis. So as a common saying, if it does not behave like a drug, if any compound does not behave like a drug, it will never be a drug. So the drug likeness of the two identified phytochemicals as well as the remdesivir were carried out using mode inspiration and employing the Lempinski rule of 5, which says that any compound or molecule that will behave like drug must not violate more than one of its rules. And the rules are: the molecular weight should not be more than 500, the hydrogen bonding should not be more than 5, the hydrogen bond donor rather should not be more than 5, hydrogen bond acceptor should not be more than 10, and the log P should not be more than 5. Going by this result, it shows that nigellidine is the best candidate here, followed by ederagelin. Ederagelin only violated one of the rules, whereas nigellidine does not violate any. However, remdesivir, which is the standard drug, violated 2 of the Lempinski rule. This is also a red flag for remdesivir. However, their molecular interaction the molecular interaction of the phytochemicals and that of the standard drug were visualized in order to see those amino acids and the type of bond involved between the phytochemicals and the target receptor. By this result, Nigella have more conventional hydrogen bond, which is stronger than every other bonds involved in these interactions. That portrays the point that Nigella is the best candidate out of all the identified phytochemicals, followed by ederagelin. And remdesivir have a number of unfavored bonding, and this is also a red flag. Those two phytochemicals, that is Nigella and ederagelin, as well as the remdesivir, were subjected to molecular dynamic simulation in order to see their behavior in the biological system. The result, the property employed in the course of the simulation, the 550 nanosecond simulations, are the root mean square deviation, root mean square fluctuation, surface accessible surface area, radius of gyration, total energy, as well as hydrogen bonding. Going by the root mean square deviation of the phytochemicals, of the identified phytochemicals, as well as the standard drug, the standard drug is represented with green color. Ederagelin is represented with the black, while Nigella is represented with a red color. At the beginning of the simulation, both the identified phytochemical and the standard drug have a root mean square deviation of approximately 0.2 nanoseconds, and the simulation, this property continues until 10 nanoseconds, where standard drug, which is the remdesivir, began its fluctuation, and the fluctuation continues until 25 nanoseconds, where it maintains the fluctuation keeps rising until it gets to 25 nanoseconds, where it maintains 0.8 nanoseconds till the end of the 50 nanoseconds simulation, whereas the two phytochemicals maintain their level of deviation at just 0.2 nanoseconds, and there was no further deviation until the end of the simulation. The same thing was observed for root mean square fluctuation, where remdesivir has begun fluctuating right from the beginning of the simulation at very high fluctuation level, which is approximately 0.6 nanometer, until the end of the simulation. This is another red flag, and it tells us that the two identified phytochemicals have better properties than that of the remdesivir, as they maintain just little fluctuation of approximately 0.2 nanometer from the beginning of the simulation till the end. However, like properties was observed, or like behavior was observed for both identified phytochemicals as well as the standard drug for solvent accessible surface area, also known as SASA, as they have similar behavior from the beginning of the simulation till the end. The same thing was also observed for radius of gyration until 25 nanoseconds, which is the same thing as 25,000 picoseconds, where remdesivir drops its gyration to approximately 2.1 nanometer until the end of the simulation, whereas the identified phytochemicals maintains approximately 2.25 nanometer from the beginning of the simulation till the end. Then the same thing was observed. Similar thing was observed for the total energy as well as the hydrogen bonding. In summary, the main properties of SARS-CoV-2 is synonymous with a gun magazine. Once it is removed, the gun becomes harmless. So, once the main properties of this virus is rendered inactive, the whole of the virus becomes harmless. So this research has helped us to understand that ederagelin and nigellidine from Nigella sativa have more inhibitory activities against the target protein than the reference drug. Also, phytochemicals from Nigella sativa has been shown by this work can therefore be explored as a source of drugs against SARS-CoV-2 and other similar infections. Here are part of the references from the literature explored in the course of this work. As stated, as cited in the course of the work, and others were kept for full publication. Thank you for listening.